Can still hear me all right?
Yeah.
And we can see my slides, everything's set up.
Yes.
Okay, great. Hi, everyone. Yeah, so my name is Mike Lee. I'm a senior scientist with Sarah's team at the BC CDC. And we have been working over the previous year to get a project up and running using satellite data to detect cyanobacteria and algae blooms in BC's freshwater lakes. And in this presentation, it's going to be broken down into kind of three parts. I'll start by giving some background about cyanobacteria, what they are, why they're a public health problem, and how they're changing with climate change. Then I'll talk a little bit about what we know and what the situation looks like in BC. And then in the final part of the presentation, I'll move into talking about where we want to go and what this project that we're working on is designed to do. So I'll start with a land acknowledgement. And I think with this kind of work, it's really important to point out that what we're doing is guided by this Coast Salish teaching, Nut Samat, or We Are One. And this is the idea that the health of humans, animals, and the environment are all interconnected and interdependent. You might have heard something similar with the idea of One Health, but really One Health is just our attempt at putting that into Western medicine and Western science, while many indigenous communities have held this perspective for a very, very long time. So I thought I would start with a quick poll question, asking everybody, what are blue-green algae? So are they a eukaryotic algae that produce a blue-green pigment known as phycocyanin? Are they photosynthetic bacteria? Are they a diatom? Are they fungal cells that colonize plants at the surface of lakes? Or are they a dinoflagellate? All right, it looks like everybody has answered. So I'll end the poll, and most people think that it is a assume everybody can see that now, the results?
Yes.
Yeah. Most people think it's a eukaryotic algae that produce a blue-green pigment known as phycocyanin. They are actually photosynthetic bacteria called cyanobacteria. They're erroneously often referred to as blue-green algae, but they're truly not algae. They're prokaryotes. They're not eukaryotes, and so it's important to understand that distinction because there are many differences that relate to how they grow in lakes. Now, cyanobacteria are found naturally and fresh in marine waters worldwide. And in those places, they're essential to ecosystem function. So in the ocean, they're one of the primary biomass producers, and they produce a significant amount of the Earth's oxygen. So they're really, really important to how our environment works. Now, they do become a public health threat when they bloom and or produce these toxins that we call cyanotoxins. In this picture here on the left, I think I'll use a laser pointer. This picture here on the left is an algae, a cyanobacteria bloom dying off in Wood Lake, which is in the interior of BC, in May 2021. And so you can see these clumps of bluish brown material across the surface of the lake, and that's actually the bloom breaking apart and dying. And this here on the right, this is a microscopic image of the implicated cyanobacteria. So, like I said, this is a global One Health threat. And with this slide, I want to point out two different and important perspectives that we need to consider when we're thinking about this problem. So if you first start on the left, when we think about cyanobacteria, when we've thought about them historically from a public health perspective, we generally focus on the public health threat posed by exposure to those cyanotoxins. And a very small fraction of the many species of cyanobacteria produce these toxins, but they produce many different toxins that have a variety of different health impacts, which I'll get into more in the next slides. And when we take this human-focused perspective, cyanobacteria generally receive a relatively low public health priority because human health incidents are rarely recorded. If we take a One Health perspective to this problem, we can see that cyanobacteria actually have some significant impacts across not only humans, but also animals and the wider ecosystem. So when they bloom, they can cause significant water and ecosystem degradation. These blooms can cause hypoxic water conditions, which cause mass fish mortality. They can block out UV radiation, which causes aquatic plant mortality. The toxins can cause wildlife, cattle, and pet death. They can reduce access to waters for cultural, spiritual, and recreational purposes among indigenous communities. Even when these blooms aren't producing toxins, they're really unsightly and they can smell really bad. So people just don't want to go to those areas when it's happening. And then they can cause a variety of food safety and security concerns, and they can have some pretty significant impacts on aquaculture when they occur around those areas. So let's get into a bit about what we know about the health impacts of exposure to cyanotoxins. So we generally think of exposure to cyanotoxins as primarily occurring through ingestion. So that's drinking water and accidentally swallowing water when you're swimming or recreating around a water body. But these toxins can also bioaccumulate in seafood. They can be aerosolized in water droplets. So if you're showering with a contaminated water source, it can actually rise up in the steam or in the water droplets coming out of the shower head and cause symptoms. And then you can have contaminated food when you irrigate crops with contaminated water or when you wash vegetables with contaminated water. And generally, what public health is primarily concerned with, because it's what we know best about cyanotoxins at this point, is the acute health impacts associated with a single high dose exposure to these toxins. These health impacts are generally mild and self-limiting, so they cause things like skin irritation, GI distress, headaches, etc. But if you're exposed to certain toxins at really high doses, it can cause some pretty severe symptoms like respiratory paralysis, organ failure, and in very rare cases, death. Now, we generally categorize cyanotoxins by their primary impact on the human body. So I've listed some here that are primarily hepatotoxins. Those are toxins that have significant impacts on the liver. There are toxins that have significant impacts on the nervous system. And then there are those toxins that just have general effects across the skin, lungs, and eyes and GI tract. Now, one important thing to know is that this is just the primary and most obvious impact of exposure to these different toxins, but all of these toxins are implicated in all sorts of other health impacts across the body. And then there are other types of exposure that can occur with cyanotoxins. So beyond just being exposed to a single high dose of these toxins, we can be exposed to multiple high doses over time or to chronic low doses of these toxins over time. And there might even be extended effects of a single high dose. So these different kinds of exposures have potential chronic health impacts that we are just beginning to understand and recognize. So these toxins have been implicated in many different health impacts, things like tumor promotion, kidney damage, liver damage, cellular damage. But in general, these impacts are fairly poorly studied. And down here on the left, this picture and this study, Holtkamp 2012, highlights one of those potential chronic health impacts that the public health community is becoming more and more interested in as evidence piles up for this health effect. So just to give you a brief overview of this potential impact, this cyanotoxin BMMA was first discovered on the island of Guam in the 1960s. And it was later associated with a unique neurodegenerative disease similar to ALS that was specific to people on this island. And people were exposed to BMMA and are probably currently exposed to this toxin through the consumption of local bat species, like you can see in this picture here. The toxin is primarily produced in the cycad seeds that you can see on the top that are produced in this tree, but the bats biomagnify these toxins in their tissues because they feed on these seeds over time. And since these earlier studies, there has been some additional evidence that has emerged over time suggesting that perhaps exposure to BMMA is associated with a wider range of neurodegenerative diseases. So, for example, BMMA has been found in the brains of people with Alzheimer's, but not in asymptomatic controls. And this toxin has been shown to be produced by many different cyanobacteria that are found all over the world. So this is an important issue for us to begin to better understand because there is a global trend towards more blooms across the globe. And there are a couple of key reasons why those blooms are increasing in frequency and severity. The first and probably the most important driver of these blooms is population growth. So cyanobacteria bloom when there is increased nutrient inputs into our lakes and waterways. And these nutrient sources primarily come from anthropogenic pathways, things like urban runoff, sewage, agricultural runoff, industrial effluent, wastewater. The list really goes on and on here. And as the population grows, not only do we get more of these pathways concentrated in certain areas, but the population is going to start expanding into new areas and potentially expose lakes and rivers that weren't previously subject to these nutrient pathways. And then, of course, we have the impacts of climate change. So that leads us to our next poll question. And I'm asking which climate change impacts are expected to increase the occurrence of cyanobacteria blooms. Is it wildfires and wildfire smoke? Is it extreme heat? Is it drought? Is it flooding? Increased precipitation? Or do we see general impacts of fires, increasing temps, increasing precipitation, or just all of the above? Like people are slowly answering here. I will end the poll. So not everyone has participated. And it looks like a lot of people think that extreme heat is important, as well as wildfires and flooding. Oh, I didn't scroll down. Yes, all of the above. That is the correct answer. All of these climate impacts are expected to have positive impacts on increasing cyanobacteria blooms. So drought, increasing temperatures, extreme precipitation, wildfires, all of these things can come together to increase the incidence of these blooms in our freshwater lakes. So I've given one example here on the right. This is a study from 2023 which looked at nutrient mobilization by wildfires. And what they found was that phosphorus and nitrogen, which are key nutrients to the blooming of these cyanobacteria, were actually mobilized in the particulate matter in wildfire smoke and then deposited in downwind lakes. And they saw a significant increase in cyanobacteria blooms in those lakes compared to before the wildfires. And then, in addition to the impacts of climate change, we also have the impacts of invasive species. So, as new cyanobacteria species are introduced into new areas of the world via human transportation on boats, things like that, they might be introduced into environmental areas where conditions were previously uninhabitable for them. But climate change is pushing those areas towards being favorable to the growth of those different cyanobacteria. And then there are a number of other impacts, one that I've highlighted here. So invasive mussels have a significant impact on the presence of these blooms because they can change the structure of phytoplankton in lakes and change the concentration of different nutrients by filter feeding. Now, something that's important to know about this issue is that the risks of cyanobacteria and cyanobacteria blooms are highly variable across different lakes, even lakes in the same geographic area. So here I've highlighted cyanobacteria concentration data from 2 drinking water reservoirs near the Gulf Islands in BC. These lakes are relatively close, they're about 35 kilometers apart. But what you can see from this set of graphs here on the right is that they have different species of cyanobacteria. And even where they have the same species of cyanobacteria, there are different trends over time in those species that we see in those different lakes. So if you look here at the first plot, this is the concentration of Dolichospermum in a small reservoir on a small island in the Gulf Islands, and then on a large reservoir on Vancouver Island. And you can see that while the concentrations in the small reservoir are highly variable and are possibly increasing over time, they're relatively stable over time in the larger reservoir. And then we expect that climate change is going to have variable impacts across different lakes. So this is data from those same 2 lakes and those same cyanobacteria species. But here I'm showing the cell density of these species against temperature on the x-axis. And if you look again at Dolichospermum, you can see that in the smaller reservoir, there's this clearly increasing association between cell density and temperature, while there's little to no association in Vancouver Island, in the Vancouver Island reservoir. So why is that the case? Why do we see these different impacts of temperature on these 2 lakes? Well, it's probably because the smaller island reservoir has a higher trophic status. So this means that that lake has more nutrients than the other reservoir. And we know from experimental studies that this cyanobacteria responds much more readily to temperature when nutrients are high. So put simply, in the larger reservoir, there just aren't enough nutrients available for this cyanobacteria to grow, even though temperature is changing. So, why is this important? I've just highlighted that cyanobacteria risks are highly variable among different lakes, and we expect climate change to have variable impacts across those different lakes. This is important because it means that we need specific monitoring in different lakes to monitor and understand those risks. So what is cyanobacteria surveillance currently look like in British Columbia? Well, overall, a fraction of BC's many, many lakes are regularly monitored for cyanobacteria. So you can see this picture here on the right. This is an image of all of the lake perimeters in BC. And you can see that there is just a massive amount of fresh water in the province. And so it's pretty much impossible to get out there and do regular cyanobacteria monitoring in all of those different areas. Now, we do have monitoring in a number of different lakes across the province, but even where those data exist, they're often disparate. They're held by many different groups. Those groups collect different variables, they use different methods, they collect it over different time frames, and they have different objectives. And so this means that really it's quite difficult to get an overall picture of this issue across the province. We only have these data sets for specific areas, and it's really hard to find all those data sets and bring them together. Now, there are a couple of provincial programs. The BC Lakes Monitoring Network does regular monitoring for phytoplankton and cyanobacteria across about 50 lakes in the province. And then BC Algae Watch is a citizen science program where the public can submit pictures and observations of blooms. And then there are a number of different lake and region specific monitoring programs all over the province. For example, we have drinking water monitoring programs in large reservoirs supplying cities. And then there are many smaller locally run programs by municipalities, water system operators, First Nations, consulting agencies, and even volunteer groups. So, given that, how many cyanotoxin exposures do you think occur in BC annually? Okay, I'll give it about 5 more seconds. All right, I'll end the poll there. And it looks like we got a variety of answers, but most people got the right answer. We don't really know how many of these cyanotoxin exposures are occurring. And that's largely because there is a limited understanding of the burden of illness because these illnesses are non-reportable. There is no ongoing surveillance of cyanotoxin exposures. And in general, they're likely to be underreported because most of the symptoms that people experience are nonspecific and self-limiting. But that being said, we do have some evidence that suggests these exposures are occurring somewhat regularly. So this graph here shows calls made to the BC Drug and Poison Information Center from 2012 to 2024 about exposure to cyanobacteria blooms. And although these numbers are relatively small, you can see that there appears to be more calls in recent years, perhaps suggesting that exposures are increasing. Most of these people were exposed while swimming or recreating around water bodies, and many different symptoms were reported, including abdominal pain, diarrhea, vomiting, headache, and the list for this really goes on and on. And one important point to point out here is that most of the people who called DPIC called in response to posted signages and warnings about these blooms. So this really highlights the importance of raising public awareness about these potential exposures. And then we see other evidence in the form of annual media reports of exposures across the One Health spectrum. So, for example, we saw thousands of fish die in a particular lake on Vancouver Island earlier this year, and this was associated with a bloom in that lake. So, in this next part of the presentation, I want to get into this question of how can we get a more comprehensive picture of cyanobacteria across BC? Currently, we are focused on water testing, and there's a good reason that we're focused on water testing. It's because these tests are accurate, they're very detailed, and you can collect as many variables as you have tests for. But as you'd expect, water sampling and testing is time consuming, it's expensive, it's difficult to scale across many lakes and time points. And in the end, even in an ideal world, these water samples only represent one discrete sampling point in a wider lake. So you can see here this satellite image of a bloom in a lake in BC. These green swirls that you can see on the right side, this is the bloom that's occurring. And this red point is a sampling location of a volunteer-run program in the area. And you can imagine that if you take a water sample from that one spot, it's really not going to give you a good picture of what's happening across this entire lake. Satellite imagery, on the other hand, doesn't require any data collection, and it enables more detailed temporal and spatial tracking of blooms by extent, severity, and frequency. So you can see with this image that we get a completely different picture of this bloom compared to what we would get with just that one sampling point. But the trade-off is these approaches with satellites are generally less accurate than water sampling and testing. So in an ideal world, what we would want is a marrying of these two approaches where we have the use of both satellite imagery and water sampling and testing. So how does this remote sensing actually work? I won't go too much into the details here, but in general, Cyanobacteria and algae produce specific pigments that reflect and absorb light in specific patterns that are distinct from one another and distinct from water. And many different researchers have made a variety of different algorithms that can detect those reflectance profiles from satellite images. The most common target is for detecting the reflectance profile of chlorophyll A, which is found in pretty much all photosynthesizing plants and bacteria. And here I've given one example of one of those algorithms called modified maximum peak height. And basically, what this equation for modified maximum peak height is doing is it's trying to detect the magnitude and severity of reflectance at this backscattering peak around 709 nanometers. So this dotted line shows the reflectance profile for chlorophyll A in cyanobacteria, and this black line is the reflectance profile for algae. And so you can see right away that the profiles are quite different for these two different groups of organisms. And so these algorithms try and capitalize on those differences, and they're built to detect different parts of these overall patterns. So what does this look like in practice? This is a satellite image and our modified maximum peak height algorithm applied to the cyanobacteria bloom that I showed very early in the presentation in Wood Lake from May 2021. So this first panel here is the true color satellite image from the Sentinel-2 satellite. You can clearly see the bloom with your naked eye. And this middle panel here is our estimate of chlorophyll across the lake. And you can see that it does a pretty good job of detecting where that bloom is occurring. Now, these methods are currently in use in other jurisdictions. So there is a pilot program ongoing in Alberta. This is a screenshot of the dashboard that they're building there. And then ECCC, or Environment and Climate Change Canada, has the EO Lake Watch program, which is applying these methods specifically to the Great Lakes. So what we are working on right now at the BC CDC is making a provincial dashboard for the province similar to what we see in Alberta and in the Great Lakes. So this is a screenshot of our early app that we've been working on. And you can see here, this is just our measures of chlorophyll in this lake, where you can also clearly see the bloom with your naked eye. So we're developing this dashboard in collaboration with partners in the Ministry of Waterlands and Resource Stewardship. And currently what we're doing is we're building our models from existing data sets. So we went out and we tried to contact as many different groups in the province as possible to get existing data sets that were out there for different lakes. And we were able to get data for about 85 lakes across the province. And we have pretty good spatial coverage at the moment for the extent of BC. So we've built our models based on that existing data. And what we're planning to do moving into this next bloom season, so the summer of 2025, is we're partnering with UBCO and Lorad Aquatic, which is a consulting agency, to collect more prospective data in specific lakes so that we can validate how good our models actually are at detecting chlorophyll and cyanobacteria. Now, this dashboard has many potential benefits. It will serve as an early warning system for resource managers. It will allow us to expand sensing to remote and under-resourced areas, areas that we can't get out and do regular water sampling. It will support evidence-informed decision-making. So should we close a lake to swimming? Should we go out and do some confirmation sampling? And we hope to work with Indigenous partners to specifically expand monitoring into lakes that have significant cultural and spiritual importance. And then from a research and surveillance point of view, this dashboard will hopefully give us the capacity to do a provincial risk assessment and start looking at which areas of the province are most impacted by this issue and which areas are perhaps the most vulnerable to climate change. And then as we collect this data over time and look back based on old satellite imagery, hopefully we'll be able to do some specific analyses to look at whether climate change has been having an impact, whether there are other local bloom drivers like population growth. And then long term, we hope to generate time series data sets to inform exposure health studies. So, earlier I was talking about the fact that we know very little about the chronic health impacts of exposure to cyanobacteria. So we're hoping that we'll be able to generate better large scale data on these potential exposures that we can use to start investigating some of those health effects. And then beyond just the impacts on humans, we hope to work with our partners to also understand impacts in the animal populations and local ecosystems. So moving forward, our next steps with this project are to continue developing those models. Like I said, we're going to be prospectively collecting data for further model validation. And then something I haven't mentioned yet is we're hoping to develop a provincial health coordination committee where we bring together partners from all of the different health authorities across the province, and then use this tool to, number one, start understanding which areas are being the most impacted by this problem. But then also for the first time in BC, we want to start coordinating the public health response and messaging about cyanobacteria and cyanotoxins, because right now health authorities all have different approaches to this issue, and certain areas that experience these blooms more regularly have developed pathways to responding, whereas other areas that haven't been as impacted really don't have these pathways developed. So just to summarize here before ending, I've highlighted that cyanobacteria and cyanotoxins are a relatively understudied threat, but that are emerging and increasing because of population growth and climate change. There are a variety of acute health impacts associated with exposure to different cyanotoxins, and there are potential chronic health impacts that we really need to start to better understand. The risks of cyanobacteria exposure and the impacts of climate change are highly variable across lakes. And so we really need coordinated surveillance in BC to begin to understand how this problem is affecting the province as a whole and how we can respond to that problem provincially in the context of these increasing threats. So I will end there and open it up for questions and discussion.